There are several ways in which materials can be transported across each cell membrane. We'll discuss a few of the mechanisms here. The main mechanisms for transport across the cell membrane are diffusion, osmosis, facilitated diffusion, active transport, endocytosis, and exocytosis. Let's talk about diffusion first. Diffusion is quite simply the movement of particles or solutes from an area of high concentration to an area of low concentration. A practical example of how diffusion works: if you took a clear glass beaker and filled it with water, if you dropped a few drops of dark red food coloring right in the top of the beaker, right after you drop the food coloring there, right where the food coloring hit the water would be very dark red. If you allowed that beaker to sit for a while, the color would begin to fade right where it hit the water, and the other areas of the beaker would become kind of a pinkish color. If you left it alone, undisturbed for long enough, you could come back and you would see that the food coloring would completely diffuse into the volume of the beaker, and the entire beaker would be a uniform light red color. That's how diffusion works. The food coloring will move naturally from an area of high concentration to an area of low concentration. This is how diffusion works. Sometimes diffusion happens through a cell pore in a membrane, and sometimes things diffuse across a cell membrane that are able to pass across the semi-permeable membrane. The degree of diffusion depends on the permeability of the membrane to the substance and the concentration gradient. What is meant by concentration gradient? Well, that's the difference in concentration on one side of the membrane versus the concentration on the other side of the membrane. So in order for diffusion to happen, there has to be a concentration gradient between the two sides of the membrane. Small molecules generally diffuse more easily than large ones. Watery solutions diffuse more rapidly than thicker, more viscous solutions. What is osmosis? Osmosis is in fact a type of diffusion. It might be helpful for you to think of osmosis as the diffusion of water. Where in diffusion, in simple diffusion, the solutes move across the membrane. In osmosis, the water moves across the membrane. So osmosis is the movement of a solvent, which is usually water, from an area of low solute concentration to one of high concentration through a selectively permeable membrane. When osmosis occurs, the membrane does not allow the solutes to pass, but it allows the water to pass. So the water moves from one side of the cell membrane to the other in order to equalize the concentration of the solute on both sides. This creates a pressure. That pressure is called osmotic pressure. It's the measure of the tendency of water to move by osmosis across the selectively permeable membrane. Crenation is a term which refers to the shrinkage of a cell that occurs when too much water moves out of the cell. That would be when a cell is in an atmosphere that has a high concentration of a solute. Water moves out of the cell to try to equalize the concentration and causes the cell to shrink. Lysis is the swelling and bursting of a cell that occurs when too much water enters in. This can occur when the atmosphere outside the cell has an extremely low solute content. Let me see if I can help illustrate this for you a little bit better. Let's see here. All right. So if we have, say, a blood vessel, a capillary, let's say. By the way, capillaries are generally about one red cell blood width wide. And we have a red blood cell. We have these red blood cells going through a capillary. Let's say that each one of these little dots represents a solute. Let's say sodium, for example. And the area outside of the cell here in the bloodstream is fairly dense, whereas the material inside of the red blood cells does not have nearly the concentration of sodium. If the cell membrane allows for the passage of water, but not freely for the passage of sodium, since there's a concentration difference here, water will tend to move from inside the cell to out. This is the movement of water. The result is that the cell shrinks and crenates and becomes ineffective. The opposite of this would be if we had a different capillary with different red blood cells, and we had a very low concentration of sodium outside of the red blood cells and a very high inside the red blood cells. Once again, the cell membrane of the red blood cell allows for water to pass across, but does not allow for sodium. In this instance, to try to equalize the concentration on both sides, water would move into the cell. In fact, it would keep moving until the cell actually swells up and bursts. Remember, that's called lysis. So in this situation, by the way, on the left, you could say that the blood, or the plasma, actually more accurately, the plasma is hypertonic to the blood cells. So these blood cells are in a hypertonic environment. In this situation, you would say that the red blood cells are in a hypotonic environment. If you had an instance where the concentration of sodium, or any other solute, by the way, were the same inside the cell and outside the cell, and there was no net water movement, then it could be said that the cells are in an isotonic environment. Now this is interesting because we have water movement here. We're talking about water movement from the cell to outside the cell, from the intracellular space to the extracellular space. But that's not the only place in the human body where water moves. This is important for us to understand as paramedics. We also have, let's see here, let's try again. We go back to the same example of a capillary with red blood cells flowing through it. And one thing they may not have explained to you in basic EMT or an EMT class, there's nothing really basic about EMT, is that at the capillary level, fluids, particularly water, pass through the capillary walls. They're very thin. Your blood vessels are very thin walled at the capillaries, unlike at the arterioles and venules. So this space out here, outside of the blood vessel, is outside of the cell, and it's outside of the blood vessel. This is the interstitial space. I misspelled it. Material inside the cell is intracellular, material outside the cell, but in the capillary is going to be the intravascular space. Here's where it becomes interesting, because now we don't only have to worry about water moving in and out of the red blood cells, we also have to worry about water moving in and out of the intravascular space. So, for example, if we started an IV on a patient and gave a very hypertonic solution so that the interstitial fluid had a normal level concentration of sodium, but we raised the concentration of sodium in the intravascular space until it was very concentrated by giving a very highly concentrated saline solution, a very hypertonic IV solution. Now through osmosis, this capillary wall will allow for the passage of water, but will not allow for the passage of sodium. So through osmosis, we can actually draw water from the interstitial space into the circulating bloodstream, into the capillary intravascular space. This seems like it would be a very effective way to maintain blood pressure, especially in a patient suffering from hypovolemic shock or some other hemorrhagic injury. However, remember that we also have this movement of water into and out of the red blood cells, so that when we give an IV solution that's very hypertonic, even though we're drawing more fluid into the intravascular space, we're incapacitating a large or a significant percentage of the red blood cells to function by causing crenation. We're causing fluid or water to move from inside the red blood cells into the intravascular space as well, and causing them to crenate and making them dysfunctional. We did a study here in this region not too long ago using hypertonic solutions to try to resuscitate patients of hypovolemic shock, and the overall result was that it was not effective, but research is still going on in this area, and I think that at some point we're going to be using varying degrees of hypertonic solution for trauma patients. Anyway, something to watch in the future.